And I skipped a whole bunch of slides there and said, oh no, I want to talk about this and this. But really, this was the big one. Okay, so when we're looking at tertiary structure, we have a lot of contributions that we need to be considering when we're looking at our tertiary structure. Okay. First and foremost, I would say we need to differentiate from secondary structure. Let's see, where can I write this? Oh, actually, we can do it on the far left, right? Because right here we have secondary structure, right? We have the alpha helix, and we have the beta sheet. Okay. By bringing alpha helices and beta sheets in contact with each other, we are then transitioning into tertiary structure. Okay. But how are secondary structures different from tertiary structure?
Secondary deals with hydrogen bonding, or tertiary deals with other kind of non-covalent interactions.
Tertiary is hydrogen bonding too. So you're sort of right.
Isn't it the location of hydrogen bonding?
It's the location of your hydrogen bonding. In your secondary structure, where's the hydrogen bonding occurring? Okay, secondary is hydrogen bonding in the backbone. Okay, well, what are we saying when we say backbone?
The peptide bonds.
Okay, this is the peptide bonds through the backbone. So if we drew out our peptide bond in purple, we've got the peptide bond. Okay, and now in blue, we have the acid, or the former acid portion of one amino acid, and in red we have the amino portion of a second amino acid, right? Okay. So when we're looking at hydrogen bonding in the secondary structure, we are referencing the lone pairs on that nitrogen interacting primarily with the partial positive on the hydrogen from your amide. Okay, those partial negatives and partial positives. That nitrogen can contribute electrons, okay, potentially out into a hydrogen bonding situation as a partial negative. And we say potentially because there's lone pairs there, and it's electronegative, so yeah, it should be potentially partially negative. But why do we say potentially and not just like, yeah, that should legitimately do hydrogen bonding? An organic chemist's least favorite word, at least as a student.
W the resonance.
Resonance. The lone pair on that nitrogen is involved in resonance. If it is involved in resonance, it cannot interact as a secondary feature. Right? So when we're looking at hydrogen bonding within the backbone, it is primarily the oxygen of our acid portion acting as our partial negative, and the hydrogen of our nitrogen or our amine acting as our partial positive. Okay? Those are our primary forces for our backbone interactions. Okay? When we move up to tertiary structure and we mention hydrogen bonding, that hydrogen bonding is now happening where? Okay? This is primarily a side chain interaction. Okay? And yes, it's a massive gray area, but that's as sharp a line as we can get. Because we could have a side chain interact with the backbone. What do you think? Secondary or tertiary? It's a backbone interaction, isn't it?
Tertiary.
It's probably more tertiary because we're involving the side chain in with that. Okay? So our difference between secondary and tertiary is very kind of slight. And if we really wanted to nail down that line, we could walk it and be back and forth across that. The most important part is that you need to be able to define those as best and clearly as possible. The biggest difference for our secondary is hydrogen bonding within the backbone, and the backbone alone. Tertiary structure will still involve hydrogen bonding, but it also involves backbone, side chains, water, hydrophobic interactions, metal coordination. I don't think it's listed up there, but I will mention it here in a second. Interactions with the solvent, okay, which is very likely to be water, but it could be other solvents if we were in a different solvent system. Secondary structural motifs. if they start interacting with other parts of our protein, all of those contribute to our tertiary structure. Ultimately, the tertiary structure is really kind of the whole protein. Okay, so if we were to look at primary structure, it's just the sequence. Secondary structure is saying, when we look at one part of our overall thing, it looks like this. Tertiary structure is when we look at the whole thing, this is what it looks like. Right? And looking at all the little pieces that interact to generate or level up from those stages. Kind of make sense? Okay, so we've got this nice figure that shows a lot of those interactions. Okay, we've got that which amino acid question, which we'll look at in just a second. But first, Anna.
So you're saying the lone pairs on the hydrogen don't interact with partial positive?
The lone pairs on the what?
Sorry, the nitrogen.
Just checking.
Don't interact with the partial positive on the hydrogen, or?
So with that hydrogen, they're already bound.
Okay.
Okay, so we would not see a hydrogen bonding interaction between the lone pairs on the nitrogen and the hydrogen because they're physically bound. But if I drew a second amine, okay, a second peptide backbone, that nitrogen on that second structure would likely not be interacting with that hydrogen. Not. Okay, so it's a bit tight down here, but let's attempt to draw one in here. Draw it in black. So if we drew something like this, oosh, it's kind of ugly. But it's kind of there. And we were to say to draw in our hydrogen bonding interaction. Okay. What you're suggesting or what you're asking is, well, is that a hydrogen bonding interaction? Mm-hmm. There is a potential for it to be hydrogen bonding, but because the lone pair on that nitrogen spends most of its time as a resonance contributor, going into the structure, can it leave the structure? No. Which means that is not the best hydrogen bonding interaction, which then sets us up, well, what is the best hydrogen bonding interaction? That was just a little bit of magic there. It's going to be between our oxygen and our hydrogen. Notice they're drawn horribly in this angle, but I don't have a whole lot of options there. Okay. Can I backtrack to a prettier image, or is that good enough?
No, I
Okay. Okay. So a fun question that could come out of this is, what are the amino acids that are contributing to each of these interactions? Okay. And I believe we talked about some. We had the electrostatic interaction between the positive nitrogen and our negative oxygen here, which looks an awful lot like the backbone. Are we referencing the backbone there? No, what are we referencing? The R groups. What R group has a negative oxygen?
Glutamate.
Okay. We have the glutamate, GLU, and aspartate. Okay. So recognizing those structures and saying where could they be involved is going to be useful. I don't expect you to have the 3-letter code for those. What I would do is provide, perhaps, might be a bit excessive, but let's say I provided a table of 20 amino acids and said which of those amino acids could match within here. Just give me some kind of labeling system. Okay, that's fair. You're just trying to recognize the patterns of what things connect and where. For the NH3, okay, lysine, which for some reason I can never remember that, but I will accept you as being correct on that.
Histidine.
The one that I remember is histidine and arginine, and I just like arginine because I always feel like a pirate. Okay? So we can get an idea of what amino acids are going to potentially interact. Are those amino acids necessarily directly bound to each other? No. That could be aspartate at number 1 in the primary sequence and lysine at number 5,000. Okay. Remember, the protein is not just a straight chain that chills. The whole thing folds up on itself. Those, as long as they're close enough to each other, can make that interaction. Okay. Quantifying the close enough, don't worry about. So ignore that. Okay. What are we getting in the next interaction? We're referencing hydrogen bonding. Okay. And again, from our side chains. What amino acid is going to give us that OH? Tyrosine's a good one. I don't know why I had to write the whole name out. I want to say serine. Threonine, yeah. Oof, jeez. I would write the three-letter. Is it the three-letter? Okay, cool. Then I can cheat my way out of that one. Okay, so it's recognizing those structural features and trying to drop them in. Okay, that top one, that one's a bit tricky. Okay, we could go through and say glutamate aspartate. Those would be valid. Okay, are there going to be others? Glutamine will work. Glutamine doesn't have a negative oxygen. Is the negative oxygen interacting in the hydrogen bond? No. It's the carbonyl. Okay? So I could still see hydrogen bonding from that molecule. It's not just saying it only happens here. We have other cases of hydrogen bonding. Okay? So we will get a lot of our amino acids contributing to the potential for that hydrogen bonding. I could have just as easily drawn for that top one and said serine. Serine is what? CH2OH? As the R group? That doesn't have a carbonyl or a negative oxygen. How does it hydrogen bond with the one in red? Whoops. Come on, eraser. There's my CH2. There's my O. There's my H. What does that O have on it?
Lone pairs.
Lone pair electrons. What's the interaction now between the oxygen and the hydrogen? Hydrogen bonding. Okay. Hydrogen bonding is a much broader category than just what's being shown there. The electrostatic one is pretty narrow. That is our only option. But hydrogen bonding is very broad. We can get a lot of different connections setting up for our hydrogen bonding interactions. Okay. Disulfide bonds. This narrows down substantially again. That will only go to cysteine. Okay, why? Okay. The only other amino acid that has sulfur is methionine, but that has a methyl group in the way. Sulfur can only have 2 bonds. If they're both carbons, we're kind of SOL as far as its reactivity. Okay. Hydrophobic interactions. Okay. Any one of, or multiples of, the non-polar neutral amino acids. Okay. Leucine, valine, isoleucine, glycine, alanine. Okay. There's tons of them that can contribute to these. Metal-ion coordination. What charges are metal? Positive. Is it always positive? Our metal ions will always be positive. The only way they can become negative is if they're bound to something less electronegative than them, which would at best be another metal. Well, that would not be biochemistry, because that's a metal-metal bond. Okay? So no, they will always be positive, which means what do they need to be bound to? Negatives. What amino acids are negative? Our glutamate and our aspartate. Okay? So yes, it's a big myriad of different pieces of possible interactions, but that's what we're trying to get at and recognize when we're looking at these interactions. Kind of make sense? Sure? Was that a defiant sure or an accepting sure? Don't worry. Yesterday in lecture, someone called me a dick. It was awesome. He thought he whispered it under his breath, and I was like, called him out on it in class. It was fantastic. Okay. This kind of goes to exam 1. This is really kind of based, you'll notice the title is saying lab people, because we used to have a lab associated with it, and it was a miserable, miserable timing issue. So I said there's no way in hell we're doing that again until it gets properly designed. But one of the labs we did was we looked at a protein that we isolated from spirulina. I'm going to take spirulina capsules, or at least heard of them. They're these dark green capsules. I think it's the Aztecs used them as a amino acid or a protein source because they have all of the essential amino acids in them. So they're pretty cool in that respect. Okay. But if you take that out, crush it up a little bit, dissolve it in a buffer, that green thing becomes blue. You have a bright, vibrant blue. Okay? Well, that blue is due to a protein that is absorbing light. Okay? What happens to that protein if we mess with it? Kind of depends. Okay? We messed with someone's shirt, well, it doesn't really seem to change much color. But proteins have their color because of how they're folded. Well, what contributes to how they fold? Those intermolecular forces. What happens if we mess with those intermolecular forces, say by adding an acid?
We change the folding.
We change the folding, which then means what happens to the color? Changes the color. Okay, so we can actually cause the color to disappear. Well, what's happening in that process is we are disrupting the intermolecular forces within the native protein, as it's called. Okay? And by disrupting it, what we end up doing is taking it to what's known as denatured, okay, or outside the natural state. Okay? All we're doing is breaking forces in the process of the denaturation. Okay? In the case of that particular protein that we were looking at, if we added acid to it, it destroys it, okay, or it destroys the native form. Could we reverse that? How? Add base. So we add a little bit of base to it, and what happens? Okay. If we did everything perfectly and we didn't screw it up too thoroughly, okay, what ends up happening is the color comes back because the protein refolds. It's achieved back to its natural state, and we get the color back. What else could we potentially do? How else could we break forces? Temperature. We could heat it up. Okay, if we heat up the protein, that gives all those bonds enough energy that they start to wiggle enough away from each other, and it denatures, and we lose the color. Okay, if we did it right and carefully enough, could we reverse that? Yeah, cool it back down, and we should get the color back. Okay, so we did a lot of things of playing with trying to mess with those intermolecular forces and see if we could change the protein structure. And it was kind of neat. Okay? Huh? Same kind of thing as cooking food. Okay, to a certain extent. We reach a point where we're no longer denaturing. Okay? What happens if I went through, say, with a pair of scissors, or in the case of my slides, an eraser, okay, and I now erase up there? Okay. Can it fold back the same way? No. The whole point of denaturing is we broke forces. What did I break?
The backbone.
Bonds, okay, which was the backbone if we wanted to get specific on it. Okay. The instant I break backbone bonds, or any bond for that matter, what happens? I change the structure, which then also makes it very difficult to fold back. I don't want to call that denatured anymore because it can't possibly revert. So what should I call it? Okay, we could look at lysis. Typically, lysis, I think, is referencing breaking the cell open, not necessarily destroying proteins. It's degraded. Okay, you've degraded the protein by breaking that apart. Okay, this is kind of on exam 1 because I think it's primarily a topic we've already addressed, looking at those forces. Okay, degrading is kind of a new concept, and those terms are kind of a new concept, but I think you guys are good enough, you can figure that out. Okay. So, if we took a look at these images, which we did really quickly, the red images represent amino acids, red versus green, right, in those two images? Right. What's the difference between the red versus the green? So we've got two differentials that we can use to describe. I would argue inside, outside first. Once we've established inside, outside as our difference, then we can start to classify those amino acids as polar and nonpolar. Because our protein is more than likely in a water environment, because it's biochemistry and all life as we know it is in water, okay, that the outside will be polar and the inside should be nonpolar. Okay? It's never that perfect, but it's pretty darn close. So that when we're looking at these images, we can then kind of project those kind of interpretations. Make sense? Which I think then takes us right back to this stupid slide. But here I actually provide the definition. Denaturation, it is easily reversible. Degradation is not easily reversible. Because I'm teaching the 130 class, let's go ahead and bring in another level on this, because we've all gone through at least that approximation. Okay. What do we look at when we're looking in their general chemistry for our changes? We rank our changes according to 2 classifications. What are those classifications?
State.
Kind of.
Physical.
Physical, you kind of have a cheat here because you've been dealing with the 130 students. The physical versus chemical. If we're looking at these 2 processes, the denaturation is reversible, which happens to match a physical change. Okay? The chemical is breaking bonds. That's our degradation. Okay? For those of you going, oh man, I got that wrong. Don't worry, almost everybody gets that wrong. Okay? It takes a little while to pick that up and nail that down. Okay? And since we said that, we might as well write that up. Okay, so we said, I already forgot. Denaturation was our physical because it is reversible. Okay, and our not easily reversible is our chemical. Okay. Does that mean we can't reverse it? If I completely destroy a protein, there's no way I could possibly ever put it back together? Okay, if I build this building and then we blow it up, there's no way we could possibly put it back together? We could build a new one that is a darn close approximation of it. Yes. So we can reverse all processes, but it's a question of how much energy we're contributing to that process. So when we're talking about our chemical, the process of reversing a chemical reaction means lots of energy going into it. Okay? Not an easy process to reverse. The physical is just kind of like, not that big of a deal. Someone spray painted the outside of the building. Okay, well, I can just put a new coat of paint on it. Okay, not that I recommend you spray paint the building. At least not in a recording. Okay. This isn't really going to show up on any exam, so don't panic too much, though I revealed the kind of big clincher on this. Predicting or seeing these structures. So when we were in organic chemistry, for those of you that at least took the year sequence, okay, how did we determine that we had the structure? Spectroscopy. Okay. In the 1 semester version, I believe you talked about IR. Yeah, so IR was used to help us identify functional groups. Okay? There are other forms of spectroscopy that can help us identify a structure. One of the big ones happens to be NMR, or nuclear magnetic resonance. Same thing as MRI. What are we looking at in that massive dot structure thing? Those of you that have looked at an NMR might recognize this. Look at the axis labels. What are the axis labels in NMR? Chemical shift, or parts per million. Looking typically at the range from 0 to 14. What's the other axis? Chemical shift, or parts per million. Looking at the exact same thing. Okay. Except when we looked at a normal NMR, we only had one axis. What have we done here? A 2-axis NMR, it's known as 2-dimensional NMR. Yippee! So those of you that had any experience analyzing an actual NMR, 2-dimensional becomes an even more fun little battle to deal with. Okay? We see signals interacting with each other depending on how close they are. Take a look at this diagonal. What is that diagonal representing? The identical amino acid. As we vary off of that, what's happening? So right here we're at 10 and 10. So on that line, we are directly referencing the exact same structure on both axes. That's why we'll always see that line pop up. As we vary off of that line, what's happening? We can see how that amino acid is interacting with the local environment around it. And then we can then predict what is happening structurally. Okay? So does that mean each of those points is an amino acid? No, each of those points is a hydrogen. So if we think back to just a simple amino acid, how many different hydrogens are there? A lot. So being able to interpret these takes a little bit of skill. That skill I don't have, which means I can't help you with. Okay? Thankfully, you're also not responsible for it. Okay? But that's what a biochemist would do, would be looking at that structure and trying to interpret it. NMR has some other restrictions to it that makes it kind of challenging. Okay, so we can run into other ways to do it. We can look at X-ray crystallography. Well, we've seen X-rays, right? You slide underneath that little thing, it shines X-rays at you, and you can see bones. Okay? Does it pick up proteins? Oh, interesting. How are we using X-ray crystallography? Okay. What we are doing is taking a perfect crystal of our protein. So you have to get enough of those molecules together to make a perfect crystal. How many of you have seen a perfect crystal? Believe it or not, probably none of you. That you've worked with salts and sugar, almost none of those are perfect crystals. Actually, diamond is typically a perfect crystal, so you might have seen that. Once we have that perfect crystal, we can then shine X-rays at it. The X-rays will bounce around nuclei. Since we're in a perfect crystal, they will bounce very particularly into a pattern. And if you look really close at that, what are you seeing? Not just a gray image, but a bunch of little black dots. What are those black dots? That was an X-ray bouncing very regularly off to the exact same point. Because we know how they can bounce through physics, we can then backtrack where it hit back to what it hit, and then we can look at each of those spots and track them backwards to where all the atoms were within the structure. That's X-ray crystallography. Okay. I kind of agree with you. I think that sounds pretty horrible too. Okay. This room is too small to say anything about it. Okay. I had friends that went through and did this. You get a PhD, pretty much, for analyzing one of these things. Okay? And you get it for varying levels, because you can go through and do this, but your resolution may be bad. Okay? You can only see a resolution where you know roughly what the atom is within 5 guesses. Okay? Well, that's pretty horrible. They have X-ray crystallographs that go down to the sub-angstrom level, meaning they have resolution of the protein structure down to about the atomic size based off of the crystal scattering. That's pretty phenomenal. Okay? How else can we do predictions about what our structure are? This then runs into modeling, okay, which is probably my favorite, even though I don't like programming, but I think the video is kind of neat. What someone can go through and do is take the primary sequence and say, well, you have all those amino acids. Okay? All those amino acids can interact via hydrogen bonding. The protein backbone can interact via hydrogen bonding. So I'm going to write a program to then say backbone interactions can do this. Okay? Hydrogen bonding or side chains can do this. And I tell the program, do it. Find the lowest energy contribution of all of those interactions and see what happens. Okay? You're looking at probably millions of interactions, and yet some crazy person can pull that off. Okay? And because I find this so fascinating, so there's our protein in its native state. So they said, this is it. Here's our protein sequence. They colored it so you could kind of see the full chain. So that was just kind of it. We threw it in the solution and said, okay, there it is. Okay? So fully denatured. What happens when they start the process? So yeah, spin it. You guys did a good job. Yep. Okay, what's that? It's an alpha helix. This is them just saying, computer, say hydrogen bondings are low energy. What the heck? And they're able to model this where they see the alpha helix start to form. And notice what's special about that alpha helix. The structure is constantly moving, and yet what's happening to the alpha helix? It stays the same. Why would it stay the same? It is an extremely stable structure. Right? That's why we defined secondary structure. We're forming something that has an increased stability. We want to identify that and label it. And now the next one's going to get a little bit harder. And, no. Come on, where are you? You ever played that? What's that? We have a beta sheet popping up. Okay, what's the issue with the beta sheet?
Loops.
Yeah, loops. How stable was it?
Oh, no.
I mean, it keeps popping up and disappearing. Why would it be disappearing? Why can't it hold? kind of accept the higher energy. Remember, our whole protein is trying to flop around and move, including the part of the chain that is not in the beta sheet. If that flops and moves, what does that do to the strand that's trying to be in the beta sheet? It's going to start to break it away. It builds enough energy that it then pops it away from all of those interactions. Okay? The alpha helix is immediately next to it, so it's a relatively easy interaction to form. The beta sheet being so distant and it now being connected with things that don't want to hold still causes it to pop and fold out of this. Okay? So kind of a neat little process. So really what I want to do now is jump to, where did you guys decide? Yeah, I don't care about all that. Stop moving it. So they ran the model a whole bunch of different times. Right? And it eventually said, okay, model, you can stop running. And they did this, looked at the images at the end of the model, and they overlaid them on top of each other. Anybody notice anything interesting about that? They overlay pretty well. Okay? Yeah, there's a little bit of mess, but for the most part, they overlay pretty spot on. Okay? If we continue this out, move on to the next part. Yep, you guys are cool. Okay, so let's get rid of go away dialogue, or just read that. Okay, the best obtained structure, right, of crystallographic quality. So they went through and they had somebody go through and do this the old-fashioned way of shining X-rays at it and then solving what the crystal structure was to get the exact structure. And they took that exact structure and they took their model and they put them on top of each other. Does that not look stupidly cool? They're like identical. This is some dude dicking around on the computer versus somebody actually taking a picture. Okay? This is like somebody saying, well, I know there's some eyes, there's a nose, there's a face, boop, and pulls up a picture of you. That's pretty cool. Okay? At least I think it's pretty cool. Okay? So that's kind of a neat little thing just to address. So we can do modeling and all that other fun stuff. Kind of a bit distracting because how many test questions would I ask you on that? Yeah, nothing. But it's still cool. Right. So now we're kind of pushing into exam 2 stuff. Myoglobin gets referenced within the tertiary section because myoglobin is a single complete protein. And when we fold myoglobin up into its active state, it is now an example of a tertiary structure. So we've got the big old folded protein, right? And notice we've got something in red here. That's interesting. We didn't talk about any red things when we looked at our structure. We talked about, what was it, a purple thing? Wow, it's way back there. Talking about a purple thing, right? We got this metal being coordinated in there. Does it have to be a metal? It could be pretty much anything. Anything could potentially fit within this. And guess what that red thing is in myoglobin? An iron would be an atom. Does that look like a single atom in there? No, it's because it's not a single atom. Okay. Iron is actually in the very middle of it, which they didn't do a nice job showing it, but is there. What is the rest of that? It's a heme. It says right there, actually. It's a heme group. Okay. Iron can bond oxygen fairly well, as evidenced by, but that involves a bunch of other stuff. Rust. Iron bonds oxygen well. That's why we have rust. Okay? So iron, if it was just chilling in our bloodstream, could grab oxygen. What's the issue with it grabbing oxygen? When it binds, the whole point of our whole bloodstream is to do what with the oxygen? Grab it and then release it somewhere else. If we just had iron atoms chilling and floating through our bloodstream, yes, they grab the oxygen, and then they don't let go of it. Okay? So we need something to help stabilize and solubilize it. So number one, the heme is there to help kind of control how that iron interacts with the oxygen. Okay? So we could have the heme floating through our bloodstream with the iron on it, and it would bind oxygen well. It would do the same thing. Except what's the issue with the heme? It also binds oxygen stupidly well. Okay? To the point that, does it ever release the oxygen? Not very well. What do you mean not on its own? There's an interesting idea. I got something that now binds oxygen. Good, I want it to do that, but I also want it to release it. Okay? How can I get it to release it? I got a dog that will fetch a stick, and it'll hold on to that stick really well.
Give it something else it likes.
Okay? I could try and give it something else it likes better, or I could just rip it out of its mouth. Give me the stupid stick, dog. Okay? What does myoglobin do? It effectively rips the oxygen out. Okay? The protein structure will change and prevent oxygen from binding quite so tightly. Okay? So this is totally animal abuse. I'm going to get yelled at for this. Okay? It's like we had that dog and we shoved something else in its mouth so that when it went to grab the stick, it could only loosely bite the stick. And I'm just going to leave it at that because it could get really bad really fast. Okay? Same thing's kind of happening when we look at myoglobin. Myoglobin binds the heme to hold on to it so it can help transport it and move it around and take it to correct locations within our organism. Okay? But it also has to have the ability to flick that oxygen off. Okay? To break the oxygen away from the iron. Okay? So the protein is there to actually stop the heme from working. Just kind of a bizarre concept. Okay? But the whole point of this is to carry it, to transfer it. If you want to transfer something, it had better be reversible. Okay? So what is heme then in the grand scheme of our tertiary structure? Okay? And that becomes a little bit of a weird thing to mention here. Okay? It's not an atom. Okay? It is interacting with the protein structure, but it's not covalently bound. Okay? We have kind of weak interactions between it. But for us to have myoglobin, we must have the heme. If we don't have the heme, we don't have myoglobin. So what is the heme then? How can I reference that? What could I call it? Yes, it's facilitating the actual chemistry there. Okay. If I lost my leg, I might want something to help me walk. What could I use? I could get a prosthetic limb. What is the heme? It's a prosthetic for our protein. Okay? So we will sometimes see these larger substructures folded into our proteins. They're known as prosthetics. Okay? They allow the protein to function appropriately. Okay? Just to give you an idea of size, okay, 153 amino acids went into this one. We had our first protein structure using X-ray crystallography, which is kind of fun. Okay? And yeah, that's pretty much all I really want to talk about on myoglobin for the moment. Okay? There's our structure of the heme on the inside. Notice we have the iron trapped by a bunch of nitrogens. Iron can bond how many different sites? 6. Okay. So what can happen is the nitrogens will hold on to 4 of those sites. The protein will hold on to a 5th, leaving oxygen for the 6th. And so what the protein can do is pull or push that iron effectively in or out of the heme, which will then change how the oxygen binds. Okay? You can also have something on the top to help push the oxygen off. Okay? Quaternary structure. This is really kind of exam 1, though there isn't a whole lot to add to this. Okay, what's happening when we move up to quaternary structure? Primary was a sequence. Secondary was how that sequence interacted with itself. Tertiary was how the sequence interacted with itself over larger distances to make a final protein. Well, if I have a protein, why do I need more structure? Myoglobin still functions. It stops at tertiary structure. Do all proteins stop at tertiary structure to function? No. Some proteins need other proteins to come together to interact with it to make the active structure. Okay? That is our quaternary structure. If we have multiple already folded proteins, come together, interact to form a larger structure, kind of like Power Rangers, right? We got the little animal things that then can jump together to form the giant robot sword thing. Why shaking your head? What's wrong with that?
Okay.
What other structure does that? You can't do that. Transformers does that. Yeah, Transformers would work. I forgot about Transformers. Was there another one? Voltron?
Yeah. Big silver thing right there.
Oh, okay, yeah. So we can build these larger structures. Right? That's what's happening with our quaternary structure. How are we going to build those? How are we going to get the different proteins to interact with each other? Side chains. Right? Beta sheets. We could have beta sheets running on one side interacting with beta sheet of another protein. We didn't say they had to be part of the same protein to form a beta sheet. Okay? Could we have alpha helices? Alpha helix on one becomes the alpha helix of another? Why not? You're right. Does that make you feel better? The alpha helix is amino acids that are
On the inside?
immediately bound to each other. Okay? They aren't necessarily on the inside of the structure, but they're immediately bound to each other. I can't move from one protein to an entirely separate protein with them still being separate. They'd be linked, which would make them one protein. Okay? Could I have the alpha helix of one protein interact with the alpha helix of another? Yes. Okay? But it's not the same alpha helix. Okay? So it's just how we put these things into larger effects, which then brings us out to this kind of fun thing, allosteric effects. Allo meaning...
So,
Does it mean cell? Oh, I kind of made up what I thought it meant. What is steric? Steric hindrance? Size. Okay. Allo, I think, goes back to change, is its Latin root. So if we have an allosteric effect, what does that mean? Changing size. When I bring proteins near each other, they can affect each other to change their size. We can change how the protein is folded by bringing them closer to each other. And this introduces allosteric effect, which we will learn to hate, probably with a passion, in the next unit. Okay? Hemoglobin, over here, exhibits lots of allosterism. Okay, so if we take a look at hemoglobin, what does hemoglobin do? Bind oxygen. Wait, isn't that what myoglobin did? Why do we have two different forms? I don't think that's the case, but there's an interesting point. Hemoglobin, notice the 4 different colors? Because it's a quaternary structure. Okay? Hemoglobin is 4 different subunits, or 4 subunits put together to form one massive structure. Okay? Myoglobin does not do that. Okay? By bringing in those units next to each other, what can possibly happen? Allosteric effects. Okay? Which means if this structure here in the bright green changes its structure, what does that potentially do to the other one? Potentially changes it, potentially changes it, potentially changes it. Okay, what is the role of hemoglobin? Bind oxygen. What was the role of myoglobin? Bind oxygen. So they have the exact same role, except hemoglobin has an extra added feature. What is that added feature? Remember, we're still binding at the hemes.
transport.
Yes, it's going to do transport. Why would it be releasing it differently than myoglobin? If we bond oxygen, are we changing the forces? Yes. If we change the forces, we potentially change the structure. If we change the structure of the protein, does that affect the other structure of the protein? Yes, which can then potentially do what?
Change the function.
Change how it interacts with oxygen. Okay? Myoglobin, since it only bonds a single oxygen and that's it, that's all it can do. It grabs oxygen and that's kind of it. What does hemoglobin do? It binds 4. So it can bind 1, but that binding of 1 can now have an effect on the other 3. What happens if I bind 2? That can now have an effect on the other 2. Okay, the question becomes is what is that effect? As Ray is already getting to, it can have a higher affinity, and that's what we're looking at when we look at a curve like this. Okay? Notice with the hemoglobin, what happens as we put in oxygen. Okay, so what is that? Y-axis is percent saturation. Percent saturation of what? Oxygen in the protein. When I have a little bit of oxygen present, what happens to the hemoglobin? How much picks up oxygen?
Not that much.
Not a whole lot. What happens to myoglobin?
A lot.
Boom. Any oxygen, what does myoglobin do?
Fully saturated.
Fully saturate, grabs it immediately. What's the problem with it grabbing oxygen immediately? Okay, if I had $5 and waved it around up here, who's going to come up and grab it first? No? I would wave $5 to see if someone just came up and grabbed it, but I don't have $5. Okay, someone's probably going to come up and grab it. Okay, I'm waving around for free. Somebody comes up and grabs it right away. What are the odds you're going to be able to get it back from them?
Probably pretty low.
Probably pretty low. Why?
Because they need it.
They already proved the energy to go ahead and grab it right away. Myoglobin grabs it right away. Can I get the oxygen back? Not quite as easily. Which means, if we think about the roles of these two, I heard a couple people already suggesting where we find myoglobin and hemoglobin. Where is myoglobin found? In the muscles, and hemoglobin is found in the blood. Okay? The whole point of hemoglobin then, if it's in the blood, is to do what? Transport, and myoglobin? Storage. Why would I want to store oxygen in my muscles?
For backup.
As a backup if the hemoglobin can't transfer the oxygen there. Right? So I want myoglobin to hold on to it until hemoglobin says, sorry, I'm out. Deal with it. And now the blood goes, or the muscles go, okay, we need our oxygen storage. Now we can start to purge from the myoglobin. Right? They have different functions. Because they have different functions, they need to interact differently with our oxygen. What happens as we start to increase the oxygen to our hemoglobin? So we had no binding affinity. Okay, what happens as I bind oxygen? It increases. Okay? But then it just runs as a straight line, right? No, what happened to it? It increased even more. So as I bind oxygen, what starts happening? I bind one oxygen, the second oxygen binds easier or quicker. The slope of that line steepens. Okay? As you bind oxygen, more oxygen binds. Okay? Until eventually it's saturated and we can't bind any more oxygen. Okay? So the hemoglobin, because of the shape of that curve, gives us some information. Okay? It has an allosteric effect. As we bind oxygen, it adjusts the structure to bind faster. We recognize that it's binding faster because the slope of that binding curve changes. Whereas when we look at myoglobin, what happens? Is there a change in the slope? No. It binds and then it stops. Okay, why would it stop? It's fully saturated. Okay? So we can look at how we're monitoring these to then predict something about how that protein functions. Okay? Myoglobin binds instantly. Hemoglobin binds in an allosteric effect. It has something changing it. What is the shape of the curve for the hemoglobin? I know it's a weird one. It's just a line. An S, known as a, for those of us mathematically inclined, which isn't really me, I just know the answer. Sigmoidal. We have a sigmoidal curve. The sigmoidal curve is represented through an allosteric effect. What is the red one? And I have no hint for this one, sorry. You just have to know your math. Or here, I'll give it a shot. Just see if we can kind of leave.
Sure.
Hyperbolic. Okay. It's a hyperbolic curve. Okay. So you're kind of responsible. I've seen it in tests where they'll say, well, this curve is more sigmoidal. More sigmoidal means they're really saying an S state, which is in referencing an allosteric effect. Okay. The protein will change its structure to bind tighter. Okay. The hyperbolic means there's no allosteric effect, it just binds. It's straightforward, on, done. Okay? So we now have ways to look at those and kind of see some relationships here. Let's go back to this, because we talked about this from exam 1, and I'm pretty sure I screwed it up. And it was kind of bothering me then, but I could think through it fast enough, so I just kind of accepted it. If we were going to look at our lungs, what's going to happen in the lungs? Okay, it's an exchange of oxygen and CO2, but are we looking at oxygen here? No. What are we concerned about here? CO2, our blood buffer. What do we want to happen in our lungs? CO2 needs to be released. So what would I expect to happen to the pH in the lungs? Okay, if it decreases, what happens to the concentration of H+? Because I said pH. Did I say pH? I did say pH.
Yeah.
Okay. I got confused with the pH, so I think we were right from the very beginning. So in the lungs, we'd expect our pH to decrease, which means the H+ concentration is increasing. Yeah, I think we did it right the first time. So if that side increases, according to Le Chatelier, what happens? It goes this direction, and we see the formation of CO2 gas, which is what we wanted. So by the way, awesome job, guys. You did that really well the first time. And it was just me being an idiot. Okay? What would we expect to happen in the muscles? Okay. We're generating CO2 at the muscles. Do we want the CO2 to stay there? Where do we want CO2?
In the lungs.
In the lungs. How do I go from the muscles to the lungs? Through the blood. Okay. Do I just want bubbles of air flowing through my bloodstream? No, I want it to be in an aqueous environment. So what has to happen? I need the CO2 to go that way. How can I get it to go that way? So we could talk about increasing the concentration of water. Okay? That's going to involve some large concentration changes. Okay? Because water is our solvent, its concentration is so infinitely high in comparison to the rest that while that technically does work, we can't change the concentration of the water. So I like where you're going, it just didn't work. So try again.
Increase the pH.
Yeah, so we got to be careful. So if we increase the pH, what happened? We decreased the H+. If H+ goes down, according to Le Chatelier, now what happens? It'll shift to the right exactly like we wanted. Exactly the same as if we'd increased the reactant, which is what you were suggesting, Anna. Okay? So we just missed, or you missed the hard one, looking at decreasing concentrations to cause your equilibrium to shift. Okay? So we now have a basis for what's happening in those systems. Okay? In our lungs, we expect to be acidic or basic? Acidic, and in our muscles, basic. Okay? That might help us in answering some really hard questions in that hemoglobin myoglobin chapter, which is officially due in a month now. Just so you know. Might be a good idea to go back and do that sooner rather than later. Okay? Just because it's fun, if I wanted to look at the enzyme that contributed to this, it's carbonic anhydrase. Is carbonic acid a good choice for a buffer? Why is it a bad choice? Carbonic acid. For those of you that don't remember your nomenclature from Gen Chem, I don't blame you. If you saw it written down as carbonic and you're like, I don't know what that structure is, you could ask. I would give that to you. Carbonic acid as a buffer, would it act as an acid or a base? Why do you say acid? It's got a bunch of hydrogens. Right? What is the issue with carbonic acid as a buffer? Well, that needs to be a product, which is also in the reaction I'm studying. That becomes a problem. Okay, remember we get that competing effect. So that goes back to the chapter 2, deciding on what buffers to use. So we kind of did a little review there. Okay, we're going to do it anyway. Okay, blood is a rather complex thing. So if we're looking at hemoglobin binding oxygen, it is dependent on a lot of different features. pH certainly contributes. The presence of CO2 is going to contribute. Why would the presence of CO2 contribute? What does CO2 do in the presence of water? Change the pH. Well, if pH is important, CO2 is going to be important. Okay? We then also encounter this BPG. Okay? BPG is a secondary molecule that can come in and change the effects of our hemoglobin structure on how it binds oxygen. Okay? And then chloride officially also. The only reason that's included there, I think, is a question in the homework, but I don't remember actually seeing it, so it may be a multiple choice. So why would we want all this dependence? Was there a difference in the pH between your muscles and your lungs? Why do you want that dependence? So checks and balances, so things move the direction we want them to. Okay, if we didn't have a lot of dependence, what happens? You get myoglobin. Oxygen binds, and I now pass out because the oxygen doesn't get released. Okay? Or the metabolism is going to have to slow down significantly to allow the oxygen levels to get low enough to then release the oxygen. Okay? My oxygen levels would have to then be very, 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 very low to release the oxygen. Okay? Which then means I can't move as much because the reason I'm moving is because of cellular respiration. Okay? I have the energy from consuming oxygen. Okay? So I have a lot of effects within it because I want hemoglobin to do different things at different points, and I want to control those different points. If I can't control them, everything turns into a mess. Okay? So what happens with H+? Well, we got a cheat here. It weakens oxygen binding. Well, look at the reaction. We have hemoglobin bound to oxygen. What happens when I add H+? Well, hemoglobin is not bound to oxygen. There we go. Our equation tells us that. CO2. What does CO2 do? Same thing. In the presence of CO2, what happens? O2 comes off, CO2 comes on. Okay? BPG, okay? Bisphosphoglycerate weakens the oxygen binding as well. Okay? We also then have the blood buffering system all contributing. So all of these things kind of stack to then change how hemoglobin is going to respond. Okay? It could respond in a way where if it becomes a weaker bind, what would happen to the curve? Let's avoid flatter for the moment, but I like where you're going. To become flatter, what has to happen? Our curve shifts to the right. Hemoglobin's still going to bind the oxygen eventually, so what really happens? I continue flat out. I don't know why I should draw it. Sorry. For those watching at home, I apologize. Okay? Flatter until eventually what happens? It starts to increase again, and it'll level off again. Making that curve, say that again? More sigmoidal. Okay? There's now a stronger effect of oxygen binding. What happens if the oxygen binding becomes stronger? It starts shifting closer to becoming myoglobin, and it becomes less sigmoidal, or less allosteric is a fair answer to, or more hyperbolic. Okay? So what we're thinking about is how do those things affect the curve and which way does it shift? Okay? So we are interpreting a graph and then overlaying tons of information. Kind of makes sense? Okay. That, for some reason, seemed to go a lot better in my head, or a lot worse than I thought it was in my head. You guys seem to pick that up really well. You guys okay with that? Okay. Wow. Okay. Fine with me. Chapter 5. Okay, chapter 5 is all about doing stuff in the lab. Is there a lab component to this class? No, why not? Because the lab component, as far as it's designed for this class, is horrible. Okay? Which is why I typically skip over chapter 5, because we just do it in the lab. But we aren't doing it in the lab, so we need to kind of talk about it. So we have a whopping 1 slide on chapter 5. Here it is. How can we take advantage of protein structure to isolate it? And this could vaguely be tested on exam 2, because it's really just applying chapter 3. How could I take advantage of the protein structure to isolate it?
Side chain, the size or the charge.
So we can look at size. Okay? The larger a molecule is, it's going to have more interactions with a surface. So we could do things like column chromatography, which is a stretch. I don't know what you guys do in lab for 230. Does column chromatography ring any bells? Okay, good. Okay, we could do thin layer chromatography. You should have done that one for sure. Okay, same idea. Okay, based on the size. Okay, is there something else that we could take advantage of? Charge. What if our substrate was negatively charged? What is this amino acid going to do? It's going to move towards the solid surface. If it is stuck to the solid surface, what is it not doing? I am stuck to the solid surface of this table. What am I not doing? I'm not moving. Okay. So I am now positioned at that part. And what does that mean? I can then make everything else move. Okay. Well, if I've got a bunch of impurities, I can make all of the impurities move and my target not. What happened? I've now purified it. Okay. It's stuck to the stationary surface. How can I get it off the stationary surface? I could try and change the charge. How could I change the charge on that structure? What happens if I change the pH? Okay. Right now, my pH is low. It's very, very acidic, okay, because everything is fully protonated. So what could I do? Add a base. What will that base do? Deprotonate. Where? Good. Okay, it'll it'll deprotonate the carboxylate. What is my structure's charge now? Neutral. What does that mean? It should have less of an affinity for the stationary phase, and it should move off. Okay. What happens if I continue to adjust the pH? If I go even further up, I could continue to deprotonate, and I could make that structure now, or that part of it, neutral. And if my stationary phase was negative, well, that's negative, negative. That's going to come flying off the column because it doesn't want to stay there. Okay? So a lot of what Chapter 5 is looking at is what happens when we change the pH. How does that affect the structure? Okay? And a lot of that centers on the neutral state. What is the pH for our neutral state for most amino acids? 5. How do you know that? Kind of. It's the PI. So when we talked about the PI, this is why the PI is important. If I want to isolate certain amino acids, I target the PI. Okay? The PI will then help me separate based off of pH changes. Okay? So if I wanted to separate amino acids that didn't have any R groups, that becomes more challenging because their PIs are all roughly 5. Okay, so how would I then separate those? That's where I could go back to structural effects. Okay, the size of the molecule. Kind of make sense? So it's neat to look at what we can do, but I don't really care. Chapter 6. Okay, so this is not, well, this slide is officially on exam number 1 because It's looking at our kinetics and thermodynamics energy diagrams, making sure we can understand and interpret it. Okay? So most everything is on here. If you take a look at the practice exam, there's a pretty big typo that one of your classmates called out. Jerk. Okay. The difference between, you're not really a jerk, you're fine. The difference between the reactant and the product, I've got labeled as G, being the Gibbs free energy. Is that the Gibbs free energy? No, what is it? It's the change in Gibbs free energy for the reaction, not just the Gibbs free energy. Okay, it's by definition the change. And as you're suggesting, Mark, in the back, is that delta G is equal to the change in our enthalpy minus T change in S. Okay? So if you look at the things that you're supposed to plug in, I just say entropy, Gibbs, and enthalpy. Officially, none of those can show up in there because it needs to be a change to show up in there. Okay? So you can fully expect that adjust to show up on your exam. Okay? The x-axis, that's not x, y. The y-axis I've got labeled just generically as energy. Officially, that's the Gibbs free energy. Okay? So if it's just listed as Gibbs free energy, then we should be saying the y-axis is Gibbs free energy. Okay? If it's listed as change in Gibbs, well, I certainly can't put the change up here. The change is now wrong because that's not representing a change. That's saying the total energy. Kind of makes sense? The rest of it, reactant, product, your transition state, your activation energy. Okay, the energy we need to get that reaction to go. Okay. The upper portion of that curve is based off of kinetics. That's the fun part that we'll be talking about in this unit. Okay. The bottom part is our thermodynamics, okay, or the energy associated with that change. Kind of vaguely? It was you that pointed out the mistake, wasn't it?
Me?
Yeah.
No.
Was it you? Waiting for the people staring at me like, yeah, I pointed that out. Enzymes. Enzymes are biological catalysts. How would that change this reaction? Speeds it up. Where is speed determined?
The activation.
The top part of the curve in the kinetics section. So it has no effect on the reactant energy, no effect on the product energy. The only thing that our curve is going to be adjusted by is that point. That point for the reaction to go faster needs to be lower. So we could try and say something like this. Okay, the issue with that is then we're still saying it's one step. Then what did our enzyme do? But for it to lower it, it must be interacting with it, which then means we have another step, at least another step. So if we had an enzyme, we really need to have at least a second hump in there to show that we had a second step occurring. Where are our steps? Each of those peaks represents a step in our mechanism. Okay. So the mechanism looks at how we transition between each point. Our reaction is how we transition to our beginning and end. What if I said that my beginning and end was now in blue, right here? Am I allowed to do that? Absolutely. Yeah. The reaction is arbitrary on what we decide. Where's our beginning, where's our end? Okay. Typically, we pick neutral states that stop doing things, okay, which is our reactant and product. Okay, but that's not always the case. Make sense? Okay. And just because I feel like it again, our next section is going to be looking at the enzyme and how it functions. So what is our enzyme? It is likely our protein, and it's how does the protein interact with a substrate. Okay? So you've probably heard of the lock-in key model, where our substrate comes in and it fits exactly into the enzyme site. Okay? That, on its superficial level, kind of makes sense, because it's kind of what seems to be happening. But that is a very superficial level. Because what does the substrate bring in? Remember, this is a chemistry class. Atoms. Brings in atoms. What does the enzyme have? Atoms. When atoms interact with atoms, what happens? We get forces. Are those new forces? Yep. If we get new forces, what likely happens to the structure? It changes. Okay? So our lock and key model, while it can work to a first approximation with a lot of enzymes, it fails pretty abysmally with others. Okay? So our other model ends up being something more along the line of an induced fit. Okay? And we can introduce other features within this. Notice that when we look at the lock and key, nothing really changes when we go back and forth between them. We have the substrate, we have the enzyme, that's it. When we move to the induced fit, what happens to the substrate? Well, it didn't really change, but what happened to the enzyme? Significant changes in the pocket where the substrate bound, right? Okay, we went all curvy to all pointy. Nice and curvy. So now death and stabby. What else changed? Ooh, interesting. We've got a change now on the bottom of that structure. What does that change potentially do?
That's the functionality.
How many people, when you have a pen drop, just bend over and just pick it up? Okay. If a pen dropped, you wouldn't all like freak out, right? Pen drops, we adjust. Why? What happens if I just bend over and pick it up? We pulled a really bad joke. What if it was soap? As the enzyme changes its interaction, that is now going to potentially change and expose different parts of the enzyme that could then interact with something else. If it interacts with something else, what can then potentially happen to the reactivity of the enzyme? It can also change. All of that we then must model and come up with the equations and mathematics and curves and fun, nasty stuff like that.